Hi, everyone. Welcome to OsteoBites. Thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Mike Lizardo from BC Cancer to discuss his work investigating the pharmacologic inhibition of EIF4A to prevent osteosarcoma metastasis. Thanks so much, Dr. Lizardo, for joining us on OsteoBites today. We are thrilled to have you. A little bit more about our guest today before we get started. Dr. Mike Lazardo is a staff scientist in the laboratory of Dr. Poul Sorensen in the Molecular Oncology Department of BC Cancer in Vancouver, BC, Canada. He completed his PhD in medical biophysics at Western University in London, Ontario, under the supervision of Drs. Anne Chambers and Ian McDonald, and his PhD focus was studying and imaging lymph node metastasis. After graduating with his PhD, he joined the laboratory of Dr. Chand Khanna in the Pediatric Oncology Branch of the National Cancer Institute, National Institutes of Health in Bethesda, Maryland, as a postdoctoral fellow. His postdoctoral research focused on osteosarcoma lung metastasis and pharmacologically targeting the ER stress response as a metastatic vulnerability. After his postdoctoral studies, he joined Dr. Poul Sorensen's research group, where he is laser focused on studying osteosarcoma lung metastasis biology and finding druggable targets for the development of novel anti-metastatic therapies. Thank you so much for joining us today, Dr. Lizardo. We are very much looking forward to your talk. And I know your paper on the study was published back in October 2024, and it has encompassed so much work. I was reading through the paper and I was exhausted just going through the experiments, just reading about the experiments that you did. So I know that this talk is a culmination of so much work. Just a few announcements before we get started. Our next virtual tumor review board for osteosarcoma meeting, which we affectionately call Turbo, is next week, March 12th at 5 p. Eastern. Clinicians are welcome to present a case for discussion, and we also welcome researchers to attend to learn about real-world clinical challenges, and I will put a link in the chat with more info on how to join that. Also, big news, factor registration just opened yesterday, so we hope you can join us June 26th to 28th in Salt Lake City. You can register by March 19th to take advantage of our early bird registration rate. Our scientific program will be announced shortly, and we have a fantastic lineup this year, including Dr. Lazardo will be there. Yet another reason to come. I'm going to put a reg link in the chat. MIB also has a conference room block at the fabulous Grand America Hotel in Salt Lake, so you can take advantage of our discounted room rate by booking at the factor reservation link, and I will put that in the chat as well. And travel award applications are already open. There are a limited number of travel awards for both scientists and osteo warriors. Scientists can apply on Proposal Central, and osteo warriors must be nominated by a healthcare provider. Our scientific travel award applications are due end of this month on March 28th, and I will put a link in the chat to find more information on eligibility and guidelines. And that's it from me. Dr. Lizardo, I'll hand it over to you to get started. And everyone, please feel free to submit any questions you have for Dr. Lizardo through the Q&A in Zoom, and we will share those with him after he finishes his presentation.
All right. Thank you, Christina, for inviting me to give a talk on this recent data that we published on uncovering a new molecular driver of osteosarcoma metastasis biology. So the basic question I've been asking actually since I was a postdoc was basically, how do metastatic cells survive in the lung? So the lung is a very stressful microenvironment. I always like showing this picture because it basically shows you human osteosarcoma cells labeled in green just arriving in the lung microenvironment. And basically, I've been studying how do they adapt in this lung microenvironment. And if we can figure out how they adapt, maybe we could develop some targeted therapy against that adaptation, preventing the cells from growing within the lung microenvironment. So what I'm going to do is basically just talk about some research from the metastasis research community and what we know about lung metastasis. And then I'll talk about mRNA translation dysregulation. And then to address the hypothesis, I'll try to link those two fields together to make hypotheses and predictions on our development of this new type of therapy. So, as some of you may know, that when tumor cells are injected into the blood vasculature, at least in experimental models, only a small subset of these tumor cells are actually able to survive at the secondary site, which is usually the lung, and it depends on the cancer type. So, cancer cells can arrive in the liver or the brain. So the majority of them will die off. And this has been demonstrated by as early as 1970, and over the years, other people have demonstrated the same phenomenon in various ways. People have known that the metastasis process is very inefficient because each step is a rate-limiting step. So from invasion to the surrounding tissue, intravasation into the blood vasculature, transit within the vasculature, extravasation, and then colonization of the secondary site. Each of those basically is a bottleneck for metastatic progression. Only a very small subset of hardy tumor cells can actually make that journey successfully. A lot of the tumor cells, a lot of this, the patterns of metastasis can be determined by blood flow patterns. So if you're in the venous circulation, one of the first capillary beds that you'll encounter is the lung. So it all depends on where the blood is flowing, as well as tumor cell adhesion to the microvasculature, and the host cells within that secondary site can also mediate metastasis. During lung colonization, over the years, people have demonstrated that tumor cell interactions with the primary or the pulmonary microvascular endothelial cells, alveolar macrophages, and type 1, type 2 alveolar epithelial cells can actually regulate metastasis formation. So here's a video actually from my PhD supervisor. He likes showing this in his microcirculation undergrad class. So this is basically a close-up view of the alveolar air sacs within the lung microenvironment. Dr. Ian McDonald is sort of a MacGyver type of scientist. I'm probably aging myself by saying that, but he can actually rig a system where he can monitor the blood flow within the pulmonary vasculature. And people, when they inject tumor cells via the tail vein, here's another example in the middle here of green tumor cells arresting in that same microvasculature. And basically the tumor cells can interact with the pulmonary endothelial cells. And so this is just a cross-section, diagrammatic cross-section of that. So between the air sacs, we have our capillary, and the green tumor cells are arriving in the lung mostly through those capillaries, and they can interact with endothelial cells as well as the other cells of the alveolar space. We have type 1 and type 2 as well as alveolar macrophages. So as I mentioned, the fate of these tumor cells that arrive in the lung, most of them will die off over time. And again, as I mentioned, this has been observed as early as 1970, where Isaiah Fiedler radio-labeled tumor cells, and then he was able to calculate how much radiation or how much counts can come from one tumor cell, or and then he can calculate backwards. If he takes a reading from the organ, how many tumor cells are in there. And he saw over time, over a period of 2 weeks, that after the injection of 200,000 cells, over time, about only 400 cells persist over time. And as well as Ann Chambers showed this in another way using a cell counting method. So a lot of tumor cells arrive in the lung after a intravenous injection in the tail vein. And then over time, you can see that these solitary cells will slowly start to disappear. And then over time, you also see multicellular foci begin to occur in the lung. And one of the main points in her lab was that these solitary tumor cells are basically a reservoir for the growth of future metastases. So her line of thinking was, if you can reduce that reservoir, you can reduce the formation of these lung metastases in the future. Robert Hoffman also showed similar data where after a tail vein injection, he saw that these tumor cells will disappear over time. And then in the Chan Khanna lab, when I was there, Sang-Hyuk Hong was a postdoc, and he showed that there was a difference between high and low metastatic osteosarcoma cells and how they colonize the lung tissue. And I provided the links for each of those studies in case you wanted to look them up. So tumor cells are dying in the microvasculature, but why? So I was also fortunate enough to work in a lab that studied lung metastasis in my master's. And what I think the staff scientist there studied was the arrest of B16F1 cells in the microvasculature. And what she found was that this physical arrest of the tumor cells actually stimulated the microvascular endothelial cells to release nitric oxide, NO, in high amounts to the point where it causes oxidative stress in these tumor cells. And they found that this cleared the lung microvasculature of tumor cells over time. And if they modulated the endothelial cell's ability to secrete that nitric oxide, they can increase the formation of metastases. Other people as well demonstrated that oxidative stress as a major determinant of metastatic colonization. Our in-house observations also show that tumor cells can adapt to this oxidative stress in the lung. For example, I have an MG63.3 in vivo lung metastasis tissue section here, where I stain for NRF2, which is an antioxidant transcription factor. And we can see here, I double labeled the paraffin section with tumor EGFP as well as NRF2. And we can see in the merge that, as well as the heat map here in the NRF2 channel, that there's a lot of NRF2 expression in the metastatic tissue. And then throughout the course of the talk, we'll ask why. So if we can understand how tumor cells are stressed in the lung microenvironment, then it's worthy to model that in vitro by stressing the cells in the same way in vitro. And seeing if they can upregulate NRF2 in a similar fashion. So we can do that with a compound called TBHQ, which is known to upregulate NRF2 in other cell models. And I'll be using this TBHQ compound throughout the rest of the talk to oxidatively stress my tumor cells. So in terms of translation dysregulation in cancer, we know that the DNA forms the template for making proteins, and then we have the mRNA, which will shuttle that information into the cytosol where proteins are made. So these are the effector machines. So it is known in the literature that mRNA translation regulation is often dysregulated or upregulated in various types of cancers. People have begun to realize that this dysregulation actually can contribute to the plasticity of metastatic tumor cells. And for example, the upregulation or the phosphorylation or constitutive phosphorylation of EI4E, which is a mRNA translation factor, can actually help the translation of pro-metastatic transcripts such as snail and MMP in a mouse model of breast cancer. In Sean Khanna's group, work by James Morrow, he found that there are differences in how high and low metastatic cells actually translate mRNAs with complex UTR. And he found that only highly metastatic cells are able to translate mRNAs with complex UTRs during the lung colonization process, whereas low metastatic cells could not. And in the Sorensen group, they have found in previous studies that YB1, an RNA binding protein, can actually translationally activate stress-related mRNAs such as G3PD1, snail, twist, and HIF1 alpha. And that's reviewed by Amal El-Nagar in this link for his review. So putting this together, I'm going to loosely borrow a diagram from Ryan Roberts' paper. So we have our many different tumor cells with different phenotypes within the primary tumor microenvironment compartment. And during the metastatic process, these tumor cells, once they move from one compartment to the next, each compartment has its own unique stresses that the cells must adapt to. And from this diagram, you can see that, again, this is an inefficient process, and only really hardy tumor cells can complete this process. So a reasonable hypothesis would be if pro-metastatic mRNA transcripts provide a survival advantage during this metastatic cascade, um, translation inhibition of these transcripts may halt metastatic progression. So this would predict that if we inhibit translation via a drug or a gene knockdown of a mRNA binding protein, we can prevent synthesis of adaptive or cytoprotective proteins against a stress like oxidative stress, and cells won't be able to adapt to that stress. So we looked at a variety of online resources, looking at patient data as well as cell lines and PDX, RNA databases, and we just interrogated which of these translation factors are upregulated. And we found something interesting in terms of EF4A1 being upregulated in tumor tissue, but very low in normal tissue. There are other factors as well. So I basically spent about a year's worth of work looking at each of the inhibitors of each of these translation factors. And when I went through this table, I basically found that inhibitors of EI4A were actually the most potent inhibitors compared to the other inhibitors like EI4E, which has an inhibitor called EFT508, or tomoversative, which is in clinical trials. It required a lot more of the drug to exert an effect on the tumor cells. EI4G inhibitors, as well as eukaryotic elongation factor, all required high levels of the drug, whereas when I looked at inhibitors of EI4A, it was in the low nanomolar concentration. And then when I introduced the tumor cells into the Puma assay, which I'll talk about later, it also resulted in low nanomolar concentrations of the drug to inhibit tumor growth. We expanded the panel of osteosarcoma cells, so various labs were kind enough to provide us with various tumor cell lines. When I moved here from the Cana lab, I brought with me the MG63.3 and the MNG cells. Alejandro Circodero provided us with his PDX cell line models, as well as we got models from murine osteosarcoma cells from the Houston lab, as well as dog osteosarcoma from Jaime Matiano's lab. So it was basically these inhibitors, or CR131B was quite potent against having an IC50 at the low nanomolar concentration. The reason why I went with CR131B, because studies have found that mice can tolerate this drug quite well, whereas sylvester can be a bit more toxic to the mice. So a little bit more about this small molecule inhibitor. CR131B is a synthetic derivative of a natural compound called flavigene, compounds that are isolated from this Aglaia species of small bush. It's an interesting drug in that it has two modes of action. So at very low concentration, it seems to bind to 5' UTRs of mRNAs that are rich in polypurine or sequences, so AG. So it basically clamps the EI4A helicase onto the mRNA transcript, preventing the scanning pre-initiation complex from starting translation. What it also does at higher concentrations is it promotes the stable association of the EI4F complex to the mRNA, thereby preventing the recruitment of further EI4A complexes. So what this is called the bystander effects, and this is where general translation inhibition occurs. And I'll show you data on that. So EI4 inhibitors are interesting in that some labs have shown actually anti-metastatic activity in the breast cancer model, as well as Dr. Long Sheng Chang from Nationwide also did a lot of work with, I think it was ROCK-A and other compounds, and they found that a lot of sarcomas were sensitive to these workoglates. And I think there were even some osteosarcoma tumors in that study. In terms of my work, I was interested to see if this inhibitor can actually modulate the metastatic process in our tumor cell models. So this is just looking at three different models, two established human highly metastatic models, MG63.3, MNG, and a PDX line from Alejandro's group. So we can see, not surprisingly, there's a dose response curve. So the cells can tolerate 2 nanomolar, like 1 to 2 nanomolar. At the 4 nanomolar range, that's where the cells can tolerate it, but not for too long. And then at 8 nanomolar, you start to really inhibit the growth of the tumor cells. So this is where I characterize the inhibition of mRNA translation. So if you incubate cells in the petri dish with actually low levels of puromycin, I think it's 0.3%, 0.3 micrograms per mil, you can actually start to label the proteins, the newly synthesized proteins. So what we did was, this is called the sunset assay. What we did was we incubated the tumor cell, MG63.3 cells with increasing concentrations of the CR131B, and we found that it inhibited the global translation at about the 8 nanomolar mark and above. And then I showed that by the same type of assay, but with fluorescent antibodies against puromycin, and I show basically the same thing, that at 8 nanomolar, we see global translation inhibition. And here I show a biochemical differential sucrose gradient centrifugation method where we see that we can measure the mRNAs with one ribosome versus mRNAs with many ribosomes called heavy polysomes, and we can find that we can change the amount of ribosomes with one mRNAs with one ribosome with treatment. It is a dose responsive, but I'm just showing 4 nanomolar here, so 8 nanomolar would be even higher at this peak. We also did some transcriptome and proteome analysis of cells just treated with the agent by itself. And since I'm interested in oxidative stress, I basically looked at pathways that were changed either up or down. And it seems as though a lot of, at least at the transcript level, pathways involved in NRF2 regulation, as well as a lot of pathways around oxidative stress and oxidoreductase activity was changed versus DMSO control. But when we looked at the proteome, we saw this kind of reversed. So it may be that the transcripts are high, maybe because the protein is low, so the cells are trying to compensate. So you can look further if see if your favorite pathways are involved. But this was done with single agent treatment with CR1. We didn't do with stress cells yet, but later on in the talk I will talk about that. Here, this is the way I mostly study or vet drugs in the Sorensen lab, as well as when I was in the Kana lab. I was using this Puma assay that he developed in around 2010 by Arnulfo Mendoza. And it's basically, we can inject tumor cells tail vein and they arrive in the lung. And we can euthanize the mouse and perfuse the lung with agarosticks, keep it in its expanded state. And then we can cut up the lungs and then basically grow them in vitro. So this is a bit slightly different from Alexander Davis's model, where I think it's a literal 2D section of viable tumor cells, but this is more of a 3D model of tumor cells growing in viable lung tissue. And this is what they look like in the Petri dish. And basically, we can dedicate a lot of lung slices to various drug conditions. And that's the utility of this assay. And we can image them at various imaging modalities like wide field fluorescence or even confocal microscopy. So we're able to do this with various cell lines, osteosarcoma cell lines, breast cancer cell lines, as well as we recently in the Sorensen lab, we developed this for Ewing sarcoma as well. So if any of you are interested, the link is here. Um, so this is just an example of what we see at day 0, looking at the lung tissue. You can see all these green dots are human osteosarcoma cells, just newly arrived in the lung, and then we can track them over time, and you can see that they can grow from day 7 to day 14. They can be kept up to 30 days, but if by that time, most of the tissue becomes acellular. So I think if your hypothesis is interrogating how tumor cells interact with viable host cells, I would stay at the 14-day time point and earlier. So we did that. We studied CR131B in this using this assay, and here I'm just showing longitudinal data of vehicle on the top here. You can see tumor cells growing to these larger lesions. But if we treat the Puma slices of the 2 nanomolar of the drug, we can see that the presence of the drug really inhibited the growth of these tumor cells in the lung tissue. And that can be quantified here in this graph. And what we did is we validated this. We validated day 14 by histology. So since the cells express EGFP, we went in with an anti-GFP. And we can basically quantify tumor burden by IHC, and we see a similar trend. Again, DMSO, and then when we treat with the drug, we can see that there are smaller lesions in the lung. And then this is for M and G cells, so we see similar results for MG63.3 cells. So then we decided to go to in vivo studies. So we looked at the effect of vehicle versus treatment with the drug at 0.2 mg per kg. We treated for 15 days and then let the tumors grow. And we saw a modest effect on the delaying of primary tumor growth, but the mice really tolerated the drug quite well. But when we looked at the lung metastases, that's where we saw a significant decrease in the number of spontaneous metastases that resulted from the primary tumor. We see similar results for the MG63 model where we did a tail vein injection. So that's injecting tumor cells directly into the venous circulatory system, causing experimental metastasis. And we saw a significant decrease of those metastases at this drug concentration. So the big question is, how does EI4A1 reduce the lung metastases? So as I mentioned earlier, that the lung is a site of a lot of oxidative stress. As you recall that one of the studies showed that when tumor cells arrest, they induce the release of nitric oxide, which is a form of oxidative, high levels of nitric oxide, which can, like in the micromolar range, which can start to cause oxidative stress in the lung tumor cells. So again, if we go back to our hypothesis, if pro-metastatic mRNA transcripts provide a survival advantage during the metastatic cascade, i.e. colonizing a site that's high in oxidative stress, then inhibition of these adaptive pro-metastatic mRNAs can halt metastasis progression. Again, if we inhibit this translation with a drug or gene knockdown, we can prevent the synthesis of these cytoprotective and adaptive proteins against oxidative stress, and that would stop the formation of metastases. So here's some time-lapse images or time-lapse videos of treating cells in vitro 2D culture with oxidative stress, TBHQ, as well as the inhibitor, even at lower concentrations. And you'll see the effects of these over time. Do you see the video running?
Yes.
Okay. So you can see that under 100 micromolar of TBHQ, the cells can still tolerate that quite well. The cells can tolerate 1 nanomolar of CR131B, but when I combine the two, you can see that really halts the proliferation of these tumor cells. And that's quantified here, where the red is the combination versus the single agent conditions. And then we see this with other cell lines as well, the M&G cell lines, as well as a PDX cell line model from Alejandro's group. So that's in 2D culture. We always want to mimic what's going on in 3D. So the 3D tumor spheroid scenario is interesting because when tumor cells are start growing in 3D, interesting things happen in terms of gradients of metabolites and oxygen and waste. So I thought it was important to model the response to the drug and TBHQ in 3D as well. So what we found in our 3D models of 3 osteosarcoma metastatic cell lines, we can see that TBHQ can really increase. So red is or the orange is basically tumor cell death. We can significantly increase tumor cell death with TBHQ treatment alone. But when we add CR1 into the mix, we can really augment that even further significantly in these 3 models. In this PDX model, it actually killed all the tumor cells when we combined the 2 drugs. If you notice with 3D, we had to use higher concentrations of the drug, and that seems to be always the case when you're growing tumor cells in 3D. They seem to require a lot more of the drug. I wanted to tease apart the signaling going on in terms of the antioxidant response going on in the tumor cells in response to TBHQ as well as in response to the drug. So with the help of one of the students in the lab, we developed this red fluorescent protein biosensor of the antioxidant response. So we basically took mCherry, which is red fluorescent protein, that's adjacent to a PES sequence. So the PES sequence will degrade the protein as soon as it's made, because we want the reporter system to be only on when the signaling is happening. And when that signaling stops, we want the reporter to stop as well. So, and this is all being driven by an antioxidant response element. So we made this. So in the absences of oxidative stress, NRF2 is constantly made, but constantly degraded. But only in the presence of oxidative stress does its binding partner KEAP1 become modified, and that allows NRF2 to become stabilized and bind to the antioxidant response elements and drive the expression of this biosensor, causing the cell to fluoresce red. So this is what it looks like when you treat cells with this reporter system over time. You can see that more cells become red, and there's a heterogeneity in response. So, I mean, that goes with this theme of tumor cells reacting differently to a particular stress. We find that there's a dose response. The more concentrated the TBHQ or the oxidative stress is, the longer it takes for the tumor cells to deal with that stress. And then we validated that reporter by looking at the protein level. So in response to TBHQ, NRF2 becomes upregulated over time, which is expected. And then you can see downstream effector genes of NRF2 becoming upregulated. And here you can see the mCherry reporter slowly being turned on gradually over time. So this is what it looks like when I treat the cells with TBHQ and in combination with the drug. So you can see the DMSO and then the treatment with TBHQ, 100 micromolar. And then you can see in combination, you can see that the cells don't like that combination a lot. So you'll see how these tumor cells respond. And here the graph basically quantifies the number of double positive EGFP and mCherry cells per field of view. And you can see that in response to TBHQ treatment, there are a lot of double positive cells. But in the presence of 1 nanomolar CR131B, we see that the combination results in basically the inhibition of that upregulation of the ARE mCherry reporter. We also did this in 3D as well, so you can see the response in 3D. And then that's quantified to the left here, or to the right here. So moving on, that's just a backup slide in case these movies didn't work. So what we really wanted to study the mechanism of action. So we wanted to do a proteomic study where we treated the cells with various conditions, 75 micromolar TBHQ alone, 4 nanomolar CR1 alone in combination. And then what we found was the resulting genes that are upregulated in the presence of TBHQ versus DMSO. And then 4 nanomolar CR1 alone versus DMSO, and then the third column is combination versus DMSO. And we can see that these are our top 10 hits here, and we see that NRF2 is actually the second highest that's upregulated during TBHQ treatment, but then is downregulated in combination. So that was interesting, and that basically confirms what we saw in the reporter studies. And then we just validated this in vitro by treating the cells with TBHQ. We see the upregulation, and when we combine with increasing concentrations of the drug, we see a decrease of NRF2. And then we looked at the corresponding mRNA, and we didn't see the upregulation of NRF2 transcript levels, but only at the end, at the more extreme conditions, we see the transcript being highly produced, but maybe as the feedback loop because the protein is not being made. And then we also looked at stability of the protein over time, and we found that stability wasn't really affected. This seems to suggest a more of a translation inhibition type process. So what we did following this was we did some click chemistry. So we wanted to acutely label newly synthesized proteins in response to these various conditions. And basically, we wanted to interrogate the synthesis of a target protein like NRF2. So we labeled the cells with this azidohomoalanine, which is a methionine analog. So the cells will incorporate this into their proteins. And then what we could do is we can switch this AHA label with biotin. So we have our set of biotinylated proteins that represents a newly synthesized pool in response to those experimental conditions. And then we can do a pull down and do a more targeted Western. So here I show under these various conditions that if I go in with an antibiotin, we see a lot of proteins are biotinylated, and then rows column 7 and 8 are basically without AHA or with a cyclohexamide, which should inhibit translation. And then from the same sample, when we interrogate for NRF2, we see NRF2 is upregulated, but in the presence of the drug CR131B, we can see it seems to go down. So again, we just wanted to confirm if the newly synthesized NRF2 is affected by CR131B. So it seems to be a very important protein. So what we did was, if we knock down NRF2, does that phenocopy I guess what we see with drug treatment? So we took some shRNAs, non-targeting control, as well as 2 different shRNAs, and we can successfully downregulate the upregulation of NRF2 in response to TBHQ treatment. When we treat with even harsher conditions of TBHQ, we can see that the cells under no DOX, so this is the DOX-inducible shRNA system, the cells can grow quite well, but in the presence of DOX, the cells start to, their proliferation starts to be inhibited. We also see this by 3D staining for cell death. We see that in the presence of DOX, which inhibits NRF2, there is more cell death in the tumor spheroids. And then we did this with Puma as well. In the presence of DOX, shRNAs against NRF2 is made, which really inhibited the growth of the tumor cells in the Puma assay. And we also did this in vivo as well. So the top row is without DOX, but the bottom row is with DOX. And we find that DOX inhibited the progression of lung metastases. And this is important because in a Korean study, they found that in a cohort of 58 patients, NRF2 expression correlated with overall survival and disease-free progression. This basically shows the relevance of what we saw in the previous slide. A pathologist in our lab, Dr. Alberto Della also looked at EF or A1 overexpression or expression in a cohort of 21 osteosarcoma patients from the COG. This is a TMA, and he found that osteosarcoma cells had a higher H-score, which is a measure of the IHC staining, and mid to low expression correlated with improved survival versus if you had high EF or A1, that resulted in poorer survival. Later in this study, we were able to get our hands on the clinical drug called EFT226. So when I do these studies, I always find I make sure I try to use a drug that's already used in the clinic. Here we basically showed the same thing that Zotatifin or EFT226, which is another type of rocoglate, basically had similar effects on our models in vitro. Again, inhibiting growth in the Puma assay as well, and inhibiting NRF2, as well as the reporter gene. So this is basically a working model of what's going on. So when tumor metastatic osteosarcoma cells arrive in the lung, without any intervention, they can adapt to the oxidative stress they encounter in the lung or other stresses. So in this case, we think they're adapting to the oxidative stress by upregulating NRF2. And then this results in adaptation and survival and proliferation, resulting in a higher lung tumor burden, which would explain why we have a greater tumor burden of the lung in our preclinical animal studies. But with intervention with the CR131B, when the tumor cells are in the lung, there is insufficient adaptation to that oxidative stress because NRF2 is no longer being synthesized, and that will lead to increased cell death and reduced tumor lung burden, resulting in a lower tumor burden in the lung. So just some ongoing work. As I said, NRF2 is an important antioxidant transcription factor, but what are the actual downstream genes that are contributing to this phenotype? So we are looking at mitochondrial oxidative antioxidant enzyme. And this data actually I'll present at MIB Factor 2025. And we're also looking at aldehyde reductases as well. They're also involved in oxidative stress and drug detoxification, as well as we're continuing our studies with EFT226, Zotatifin, and we're looking with drug synergy with second-line chemotherapeutics, such as gemcitabine. And we found that there were additive effects with gemcitabines. And interestingly, we found that there seems to be a pro-ferroptosis signal when we treat the cells with EFT226 and oxidative stress. So these studies will be ongoing. And then with that, I would like to thank the team in the Sorensen lab. Again, Dr. Sorensen, for allowing me to ask these interesting questions. Chris Hughes was the bioinformatician / biochemist who helped with the omics studies. And then we have Yozu Hang, who helped in the animal studies as well as helping in design. He's a gene whiz in making plasmids, so he helped me make the plasmid, as well as the pathologist in the lab, Alberto Della and Tara Ship, who also helped out. Again, this would not be made possible with other people in the lab. Our collaborators, Jerry Pelletier, who first contributed the drugs to our studies, as well as Alejandro, Leanne Sales, and Alex Lee at UCSF, as well as Effector for providing us with this drug, as well as the BC Cancer Foundation and Axel Lemonade. So with that, I'd like to wrap it up. Do you have any questions?
Fantastic. Everyone, please feel free to put questions in the Q&A for Dr. Lizardo. And I just want to thank you for that incredible summary of such a comprehensive, 360-degree interrogation. I like just kind of the layers of the experiments, and also just, cross-validation across all these different experiences really kind of helped tell that story. So thank you so much, because I know that was an enormous amount of work going into all of that. So I might kick it off with some questions. And I I wanted to start with like a really basic question, which is, I mean, you kind of addressed this, I think, in a couple of your first slides about just even why osteo metastasizes to the lung. It's such a harsh microenvironment, kind of what makes it such a favorable destination. And I think you had mentioned that it's mediated by blood flow pattern, but I just wanted to understand that a little bit better. And it's just kind of like top destination for blood flow, and that's kind of why they end up there.
Well, it depends on the cancer type, but if tumor cells escape into the venous circulation, one of the first capillary beds that they can arrest in is the lung capillary bed. And then basically it kind of filters the tumor cells from the blood. But that depends also on the size of the tumors. If the tumor cells are small, like in Ewing, at least in the models that we observed in the Sorensen lab, they can shoot right through the lung and end up in other places in the body. So it really depends on the size of the tumor cells, the size of the vasculature that they end up in. But also, I want to add that adhesion interactions can also influence how tumor cells arrest. So, for example, B16 melanoma cells can really adhere to the wall of the portal vein in the liver if the liver is stimulated with some sort of inflammatory agent. So it really depends on the context of the type of tumor cells that you're looking at. But again, it goes back to that, the right seed and the right soil in terms of the tumor growth and the distant site that it can grow in.
Gotcha. We have a couple questions that have come in. One is, since this intervention blocks the very early stages of the ability of cells to survive in the lung upon arrival, is there a sufficient window for treatment with the drug shown today?
So, I mean, if I were to design, designing clinical trials is a very complex thing, and Katie Janeway has said a lot about that. So, I would say, in terms of clinical trials, it would be we could combine Zotatifin with a second line chemotherapy like gemcitabine or other agents. So we know. I remember during my postdoc work, it was drilled into our heads that when tumor cells can get into the lung and they can stay there as single cells. So in chemotherapy, yes, does reduce that pool of single cells, but if we can eliminate that pool even further by using Zotatifin to get those cells at the single cell level, I think that's where we can see a signal in terms of really hammering the tumor cells early on can result in downstream effects. So if you get rid of the tumor cells and when they're single cells in the lung right away, I think that would prevent reduce the reservoir of tumor cell metastasis that can form in the future. That makes sense.
Yeah, so follow up actually to that, because I was kind of interested in that as well as obviously the clinical translation. So what about adding it to upfront therapy versus second line chemotherapy and to prevent the metastasis, and if that were possible, is there a biomarker to determine which patients might benefit the most from that?
So from what I read in the literature, a lot has been done to combine four agents, additional agents to the MAP therapy, and it doesn't seem to improve outcomes. So I'm not sure if I would add it to MAP therapy. As for a signal, what people have done was look at circulating. It can be either circulating tumor cells or even circulating other types of cells. Like you could probably look at immune cells to see what genes are downregulated in response to the circulating drug. So this drug is known to downregulate c-Myc as well as other genes, and those have been used as markers of drug activity. So if we can isolate lymphocytes from the blood we can see whether or not c-Myc or other recombinant targets are downregulated. So that I would use that as a biomarker of the activity of this drug.
Okay. Couple other questions. Thanks for the presentation from Dr. George. I thought Effector Therapeutics stopped their trials with their Rocaglate inhibitor because of toxicity. Is that not the case?
I think they stopped it because of the EI4E inhibitor, EFT508, and I think the results that they got wasn't as good as they expected. So I think it was due to that, because I think EFT508 or tomofersetab was their lead compound, and then this EFT226 or Zotatifin was their other compound that they were studying. So I think it was more due to that clinical trial with EFT226, the EF4E inhibitor. As far as I can tell or have read, that Zotatifin was well tolerated in patients, and they identified a maximal tolerated dose, and they went forward with phase 2A trials. I think the trial, well, now the company I think is defunct. I think those trials are wrapping up, and I heard that it might be bought up from another company, but who knows? These things change.
Okay. Another question, compliment, very nice presentation, great work. Did you try subcutaneous and orthotomic for metastasis, models for metastasis?
We didn't do subcutaneous because we wanted to stick we wanted to stick with like so the models that I used were leg tumor models. So we wanted to grow the tumor near the bone and have the leg tumor grow. So short answer is no, I didn't use it in subcutaneous model. What was the other model?
Orthotopic.
So orthotopic. So we injected it not into the bone itself, but just around the bone.
Gotcha. And actually follow up question on that, because it looks like, I think you had a slide where you said that in both the tail vein and the orthotopic models had the CR131B inhibitor had same effect. It inhibited metastasis, which is great. But I am wondering, like, when you are studying, like, it seems like there might be additional stress from metastasizing from bone to lung versus just, going from bloodstream to lung.
Yeah.
And so did you see any kind of differences there? I mean, the end effect, it sounds like it was the same. It was able to inhibit mets in both, but
yeah.
I'm curious.
So in one model, the MG63.3, it actually really significantly inhibited the primary tumor growth. So that was good. And then so that was a primary tumor growth model, but then we did an experimental model of metastasis, and it really inhibited experimental metastasis using the MG63.3s.
And actually, can you expand a little bit more on that? Because as I was going through, like, there's, I feel like, and maybe, I don't know if it was in the slide, perhaps it was in the paper, that there was a mention of a cytotoxic effect from the CR131B. Though I know in this context, you're talking about it more as inhibiting growth, blocking proliferation, preventing metastasis. But can you expand on that a little bit more?
So CR1 was one of the better tolerated recoglates in mouse studies, at least. Other recoglates that I studied, like Silvestrol and other compounds with weird numbered name, the mice didn't tolerate those. So there's something about the CR1 structure that the mice can tolerate well. And I suppose that Zotatifin or EFT226 was also well tolerated in mice and in humans. So I think it depends just the nature of that particular recoglate. And its chemical structure is, I think that's what determines the toxicity. So, for example, there was a dog trial looking at beagles. The beagles didn't really tolerate Silvestrol well at all, and a lot of the dogs, I think, experienced a lot of either toxicity or death from Silvestrol. So. But other recoglates, again, like Zotatifin and CR1 was well tolerated. And there are up-and-coming ones as well that are synthesized by collaborators that seem to be well tolerated. So I think it's really due to the nature of the compound and its functional groups that determine the toxicity.
Another kind of follow-up question on this. When you injected around the bone, did you see metastases?
So when we injected around the bone with M&G cells, over time, about a month of tumor growth resulted in spontaneous metastases. So, I didn't do an intratibial injection. I did what we did in the Chow Canal lab was to inject periosteally, like around the bone.
So I think you talked about there being this correlation. You showed some nice charts that showed a correlation between the EIF4A1, which you have to really come up with a nickname for that.
Yeah.
And the CR131B.
Yeah.
Yeah. Because you don't say that all the time, do you? That's a mouthful. Okay. But anyway, so there seemed to be a correlation. I'm just going to call it EI for expression and prognosis. Was there a correlation in expression and response to the inhibitor as well?
Just trying to think. Well, so the models that I studied, the MG63.3 and the MNGs, were an interesting model to study because we have RNA-seq data from those cell lines, and they had high levels of EI4A1, so which is why I basically studied those cell lines versus their low metastatic counterparts. So there may be a correlation in terms of the level of EI4E or A and the response to the drug.
Yeah. And I guess the other thing, we had talked about this a little bit about like a biomarker response, and you had talked about c-MYC, but because this is like, so it's not like all osteosarcomas had this kind of overexpression, right? And so an oxidative stress is something that kind of is something that regardless of gene expression or something like that. So I'm just wondering, like, percent of the population of patients, that would benefit from this, or is there kind of a subpopulation where this is kind of really the pathway that the osteosarcoma cells are relying on to kind of get around and to metastasize? Are there osteos that are reliant on other pathways?
Well, I'm sure there are, but so if you recall, the drug is an RNA helicase clamper.
Uh-huh.
So it causes EI4A1, which is a RNA helicase, to clamp onto the transcript. So if you can assume if you have high levels of EI4A1, I would say there's more inhibition of the translation of those transcripts that have those UTRs that are exquisitely sensitive to recoagulates, like that have high polypurine sequences. So you can imagine if you have a lot of EI4A helicases that are clamped onto pro-metastatic mRNAs, that they will no longer be able to synthesize those pro-metastatic proteins. So I would hazard a guess, an educated guess, that there will be a population of patients that can be stratified according to their EI4 that can respond to the drug. That can be based on the expression levels of EI4A. Mm-hmm. And as you saw from that diagram with the TMA from the COG, that there's a spread of expression levels of EI4A.
And since I think you also had a slide where you were showing that you could IHC stain for that, so is that something that could be used as a way to determine if someone would be a better candidate?
I mean, in terms of if they had, like, we did IHC on their biopsy, mm-hmm. I think that would be an interesting pathological study. Maybe that's something to pursue with the COG.
And then I'm going to sneak in one more question before we have to let you go. So what about so I think and I know you're, especially with the setting that you're talking about, you talked about like in clinical translation, potentially doing this, combining it with second line therapy. And I'm wondering if it's possible to kind of look at in vivo studies with pre-treated cells that have been pre-treated with MAP, because certainly that's like another layer of stress that they're going through. And so wondering how those pretreated surviving cells might be different from like treatment naive cells.
Well, interestingly, NRF2 has been associated, or its downstream proteins have been associated with resistance to cisplatin and doxorubicin. So, if CR1 can inhibit the synthesis of NRF2, I think you'd probably target the cell's ability to be resistant to drugs like cis and MAP. But you said after MAP treatment, right?
Well, actually, what you're just saying is really interesting because would that be maybe potentially a reason to combine it with MAP?
It could be, but I mean, I would defer to people like Katie Janeway in terms of designing such a trial.
Yeah. But that's actually really interesting on kind of sensitizing resistance. But I was just thinking it was just interesting kind of being experienced, just to add to the other, 10 million other experience that you did, looking at pretreated cells, just since they may have, different like adaptive stress.
Yeah, I think what you asked is sort of what we're trying to ask in the ongoing study. So, we're looking at the transcription factor NRF2, but it's downstream genes. So what are these genes and how do they affect response to chemotherapy? So I think those are ongoing right now. Yeah.
Okay, well, thank you so much for this fabulous presentation, Dr. Lizardo. Thank you for joining us on OsteoBites today and for making it better for osteosarcoma patients. More information on this and all OsteoBites can be found on our website at mibagents.org and on our YouTube channel and at your favorite podcast place. And then we hope to see you next week on March 13th. We're going to be joined by Dr. Matt Dietz from the University of Utah. He is going to be discussing optimizing Ewing sarcoma and osteosarcoma biopsy acquisition, consensus recommendations from the Children's Oncology Group Bone Tumor Committee. You can find our OsteoBites lineup for the next few months on our website. If you ever have any topics that you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you so much, Dr. Lazardo, for spending an hour with us today. And thanks to all of you for taking time out of your busy day to join us. We hope to see you back here next week, March 13th, when we chat with Dr. Matt Dietz. And Dr. Lazardo, look forward to seeing you at Factor.
Looking forward to seeing you in person.
Thanks so much.
Thank you.
All right, bye.